And so part two, which even has a different name in the program, let's talk about what happened with QuickMD since this original version. So a lot has happened. We created a QMMM interface to it, so you can now do also hybrid QMMM simulations. You can run a classical simulation and then simulate QM. You can look at orbitals. You can look at chemical reactions. You can very easily select what part is going to be QM, what part is going to be MM, which is something that there's no other code that does it. We also did a model factory integration, so you can design your binders like your molecule right now. You can also change a molecule that is there in case you want to use in your MD simulation. For instance, you crystallize it with one drug, but then you actually need to study something that is slightly different. You go there and change the atoms. So that's relatively simple. We also did an automated force field parameter search using CGNFF. Like if you have a drug or anything like that bound to the system, like a cofactor, you can do that. And there are many, many other tools that were implemented into QuickMD. Specifically about the QM part, it's pretty cool that now you can actually do things like this. You know, like you can have molecular orbitals, you can do the QM simulation and analyze it. You can analyze each one of these orbitals and see how they evolve over time. So you can see the energy that you have in the occupied and non-occupied molecular orbitals, and you can look at that as like a quantum chemist would do. You can at the same time generate a very nice image like the one you're seeing in the left-hand side here. How much the VMD users actually use QuickMD? We asked that question early this year in the VMD survey, and the answer was that about 8.5% of all the VMD users use QuickMD to prepare simulations. It might not sound like a large number, but remember, there are a few things you have to take into consideration. VMD users, like the one that was just talking to me right now, the Lara, she's a GROMACS user. You know, like a lot of the people are actually GROMACS user, Ember users, and LAMPS, and all the other codes. And so QuickMD only prepares simulations for NMD. And second is, there are other tools very powerful out there, like SharmGUI, which is great for, especially for simulating membrane systems and things like that. So QuickMD actually has quite a lot of users if you also consider that VMD has more than 100,000 users, so which is a pretty good result. So we're very good that QuickMD in such a small time is growing so much. That kind of helps, like, incentivize us to go for the next version. So we are working right now on the QuickMD 2.0, which hopefully will be part of a VMD 2.0 that we hope to release in the next couple of years. The new upcoming features of QuickMD are being developed by a postdoc in my group, Diego Gomes. Other than the fixing the bugs, which is always there, right? What do we need to make for QuickMD better? And that's like a better version of QuickMD. And the part that we decided that was the first thing we should tackle was we need to do better, like, and that's for VMD and QuickMD, everything. We need to do better structural preparation. And so the first idea was to integrate it to Alphafold, which is this new big thing using AI for folding. We actually developed already this first interface, and we are calling it the Quickfold, which is connected to QuickMD, but you can also use without. Quickfold is a new model interface for VMD and QuickMD. It connects, like, you basically go there, say the name, an ID, say a name, and type a sequence, and you're going to get a structure out of it. At the end, we got a structure that was created using Alphafold, using artificial intelligence to find this. So in this case, here's a pretty simple one, it's like alanine, just 10 alanines. That's no big challenge there, and it works quite well. Quickfold works in the following way. You come here and you give a name, an ID for the job. You are going to have to tell where the Quickfold or the Alphafold is installed in your computer. There are tools out there right now that use online versions, like that you can retract, like you can retrieve the structure from them. However, that limits a lot what you can do with it, like in terms of size and also like in time. So we decided to first create a version that was running in your own computer. You just need to install Alphafold, which is not that complicated. And then in a second version in the future, we plan to have our own server to provide that also like as a online tool. You're going to have to say what kind of database. There are a few options of databases that include in the Alphafold. These are all Alphafold kind of input information, and you need to tell where is the where is the output is going to be. Then you come here and type a sequence, and then you click load the sequence, and you're going to fold the structure, and that's it. You fold the structure, and you can open it. And right now we're working on the second version of it already that is going to have also some analysis tool on it. What else do we want from QuickMD 2.0? In terms of better structure, we wanted to create also a homology modeling interface, a small molecule designer, like a better manufacturer, a docking software, all of that we want to integrate into QuickMD. Maybe create a quick dock. We also want to improve our membrane module that can only do a few kinds of membrane right now. We are working with Emad and Separate in his group to do that right now. And the idea is to have a very complete kind of membrane interface there that you can select basically any kind of membrane lipid that is available. If you have parameters, you will be able to do. That's the plan. We want to have an automatic protonation state detection, and we also want to include some more advanced protocols, for instance, for free energy kind of calculations. We want to be able to do graphic exchange, CoVars, wide sampling, free energy protocols, all of that together. We also want to be able to prepare simulations using other MD software, like that's something that was asked before. There is a way right now to convert NAMD files into GROMACS files inside VMD. That's like Topo tools, and we want to use that to convert it to Topo tools. I think it was. And we also want to better integrate it to NAMD3. Right now, when you prepare it, assumes you're using NAMD2, but we also want to integrate that better with NAMD3. And last, we also want to, with Mariano, who is here, TopoGROMACS, somebody mentioned here, might be that. Mariano, who is here also today, we want to revamp this analysis interface that Mariano is actually working on right now. If you have any suggestions, also, you feel free to contact Diego, who is also here today with us. And also, if you have any questions to me, I'm here to answer right now. So actually, Diego has something to show. He's going to show you a live demo of the Quickfold. And that was it. So this is the Department of Physics here at Auburn. If you want to visit us, we'll be happy to have you in town, or if you're around. Diego, can you share your screen? Is that possible? We cannot hear you in case you're talking.
Okay, can you see me now?
I can see your screen, but not you.
Oh, wait.
I can see you too.
Camera was covered.
Okay. Oh, we have a question first. Just 1 second, Diego. We have a question. Delara, can you?
Thanks a lot. I wanted to know, I saw that the life will be easier for the ones that want to do simulation with it, with this QuickMD. I want to know, is it easier now, for example, to make a PSF file with QuickMD, or this step hasn't changed?
What step? I didn't
understand. To make the, for example, PSF file, to make the topology file, force field. I want to know if is it easier with QuickMD, or it doesn't do anything for the force field.
I don't know how you're actually doing to tell you if it's easy or not, because there is a very easy way in VMD. But like in QuickMD, you just tell the protein and you get the PSF. You don't need to select anything. It's automatically. But there is a way of doing that in VMD already with the auto PSF tool.
Oh, so, because for example, with when using GROMACS, the challenging part is to have a, for example, a good topology, or I mean force field file. I want to know, for example, with QuickMD, is it easier now to have this PSF file in comparison with the, for example, using NAMD in traditional way, I mean?
So, okay. So
Hasn't anything changed in this respect
there
or
are
not?
ways you can get a PSF file, and maybe let me explain that way.
Okay.
There is a very crazy way of doing it by hand, which is step by step, which I don't know a single person that ever did that, but maybe there are people out there doing it. You can use PSFgen, which, like, I'm talking about NAMD specifically. PSFgen is where is a script. You tell a few things about your system. You say what the PDBs are, and it generates a PSF. That's how most of the time I do it. There's auto PSF in VMD, which you basically load a PDB and it does everything for you. There are very few things you need to select. It's all automated.
Okay.
And VMD, it doesn't even know there's a PSF. You know, like, you're not even going to be aware of that thing. Like, it's in the background, it's there. It's all done for you without you even notice. So yeah, it's easier in that sense, but it's actually using this auto PSF too, which is an automated version of already in VMD. So okay, so it's not a complicated thing is in NAMD to have it's a PSF. It's not a complicated thing in NAMD for at least 10, 15 years, I would say. Okay. And this PSF file contains all the, for example, interactions or not? Or there are in another file? I mean, for example, bonding interactions, dihedral, all of this information is in PSF file? Yes. Okay. And do you need sometimes to change some parameters according to the literature, or the parameters are trustable? For example, the force constant, something like this. They are pretty reliable, but there might be a reason for you to change one of them for some reason. You know, like, it's there are cases where you need, like, you might be biasing the system into a conformation that is not so standard and then you need it. But in that case, you can either change in the PSF directly, or you can get another force field parameter file and then create your PSF based on that force field parameter file. So, so that's getting to the more advanced kind of user, but it's simple. It's not too complicated. So most of the time, the PSF that is automatically made is fine for the simulation. Most of the time. Yeah, sure. For 99.99% of the cases, yes. Oh, so that's great. Thanks a lot. Thanks. No problem. Other questions? I think the order was Dilara, then Sonal.
Yeah.
So, sorry if I see your name wrong.
No, it's okay. I just have a follow-up question.
Yes.
So, talking about collagen proteins or peptides, do you think I could use Quickfold to develop their structures?
I'm pretty sure you can.
Oh, okay. I'm going to try it then. Thank you.
Yeah. Next question is from Som. If I'm saying that right, I hope.
Yes, sure. So my question is follow-up.
Sorry, I cannot hear you. It's very, very low.
Oh, sorry. Now is it okay?
It's very low, but I can increase the volume here and see.
I have increased the volume. Is it okay now?
It's still the same, but you can talk. I can hear you.
Okay. The question is regarding the same structure forming the protein having the iron complexes, for example, iron or metal ion. So this Quickfold can prepare the force field file of that iron also, or that complex also.
I'm gonna let Diego answer that one because I don't know. As far as I know, no.
Because in Gromex and other these things are not available, that the force field of iron.
No. That's a different question. You're asking two different questions. Like, the number one was if you can fold. If you can fold, I don't know, because that's one question. Like, in terms of parameters, yes, they are available.
Okay, so in this case, in Quickfold,
if you have the structure, QuickMD can handle. If you have the structure, parameters exist.
Okay, if parameter exists, then only it can create a file, force field file means.
Sorry?
If parameter are available, then only it will create a force field file or structure.
The force field file is an input, is not an output. Like, it's gonna create a PSF file that is based on in the force field that is available, like charm force field.
Okay. Another question, the pH it is included in that we can include pH or something like that?
We don't have yet an automated version for calculating protonation states, which would be the pH, but it is something that is it's kind of in the future that we wanted to implement.
Okay, thank you very much.
No problem. The next question was from Pete.
Okay. Hi, Rafael. Just a short question. In the current iteration of QuickMD, is it also already possible to do replica exchange, electrodynamics, or not yet?
Somewhat. It's not automated, so like it's gonna generate all the files that you can use in a replica exchange,
Okay.
but it's not. You're not gonna be able to just start. You know, like you can basically generate everything there, and then you're gonna have to adapt the config file to run the replica exchange.
Okay. Okay.
It is part of our plan to make that easier, but it's not there yet. But like the
Okay.
changes are very small. Like after having QuickMD preparing everything for you, you can even do the equilibration, all of that, and then when you go for a production run, which is going to be the part that is going to be parallelized, like in replicas, it's relatively simple.
Okay. Thank you.
So if we don't have more questions, I'm going to let Diego give a quick demo. So we're talking about Quickfold.
Thank you, Rafael. So I'd like to share a few more details about Quickfold that we have so far. So it's still in beta, still in development, and I think it was released on the current version of VMD. So this is the version I'm going to show you today. So Rafael already showed this to you. So basically, Quickfold is a simple interface for Alphafold. So you don't have to go through all the details of the Python scripts and all the complications that are involved in making it run. So basically, in the interface, you're just gonna give like a job ID. So it's a unique identifier for you to run. Well, you have to give a path to where you installed Alphafold. So with Quickfold, we have a small script to help you install it easily, pain-free. Well, there's something you cannot escape, is that you have to download the Alphafold databases right now. That is really painful because you have to download about 2 terabytes of data. And then you have to configure your output. Then, I mean, it is simple. Just put a sequence or load a FASTA file and write the configuration and fold. I mean, that's the whole point. So fold will get the FASTA sequence, run a multiple sequence alignment against like a huge databases that you probably downloaded, and then find template and find information in the alignment to feed the Alphafold model and build the models. So when you're done doing the sequence alignment and running and building the models using the parameters, you just load and align to see what you got. So a basic installation scheme and what you need to do to make it work. So first of all, since it's Alphafold is Python, it is recommended for you to create a new Conda environment. So Conda was the package of choice. And the reason is that Alphafold doesn't work by itself. So it needs a lot of extra software. So using a Conda environment will help you install it flawlessly. When you've done that, create an environment, you have to download Alphafold from the GitHub. I mean, it takes a second. Install all the required software. So there's a script that helps you install the libraries and the multiple sequence alignment software. And in particular for Quickfold, we're gonna need just BioPython to do it. And then the painful part right now is that you have to download all the databases that Alphafold needs. So if you want to use the full model, you have to download around 400 gigabytes, and then you extract, it gets to 2 terabytes. If you want to use the reduced set, it will be just 400 gigabytes. I mean, it's a lot, and we're gonna work on that in the future. So what are the expectations for you to run the software? So Alphafold does 3 things. So compute the sequence alignment to get the information. from that information, infer a new model for your structure. So model inference is in the beginning, and once it got like a initial model, it has to do some relaxation. So what should you expect depending on the size of your protein? So first of all, to run the multiple sequence alignment on these huge databases, it doesn't matter how big your protein is so far. So I tested from 90 to 900. So it doesn't change a lot the time it takes. So around 15 minutes, 12 minutes. Second, and that will really depend on the GPU you have. So to infer a model, so it has to read its model parameters and then use a lot of computations that depend on the system size. So when I go to 90 to 900, it changes a lot, almost linearly. And finally, and this also will depend on the hardware you have, especially the GPU, is the model relaxation. So it varies a lot depending on the size. But something interesting is that some proteins, I mean, it's not linear. I mean, some proteins take more time to relax than others that are bigger. So there's really case dependent. So to tackle some of these problems, especially downloading the database, we are upgrading the Quickfold that is not on VMD right now with some information that Rafael told you. So the most important in this sense is the run mode. So we have developed an API so you can submit your calculation to a supercomputer. So basically what you're gonna need on your computer will be the FASTA sequence. So that occupies almost nothing. And then you can run the calculation elsewhere, and also you don't have to download the databases. You can pick the one you prefer that will be in the supercomputer or on the remote computer that you want to use inside your lab. And you don't have to keep downloading it on your own laptop or weak computer. If you have like a super expensive GPU on your computer, we implemented a split mode. So you can run the multiple sequence alignment elsewhere and then do the folding in your expensive computer. And also we are adding some analysis tools to the whole workflow. So basically what is missing right now is how to evaluate the quality of your model. So I'm showing one of the measurements, that is the alignment coverage. So you give a sequence, you see from the templates, how many templates can I find along the sequence that can give me information on how to build the model. So a simple example, this ubiquitin that everyone uses. I mean, you can find in the templates lots of sequences that can give information on how to fold it. And then another example is the exokinase, which is a huge protein, with that one with the 900 residues. So the two extremes, you find way less sequences that can cover this sequence to give you information on how to build it. So in addition, we are implementing the standard metrics for the CASP experiment to one of the standard metrics to measure the quality of your folding. So the LDDT. So there's an estimate from 0 to 100 of how good is each of the residues in the prediction. Also, something that is used a lot is a distance map. And well, inside VMD, you can do multiple sequence alignments. And so you can visualize and inspect all the alignments that Quickfold or Alphafold propose to you. So you can investigate if you have problems in some areas of your model, go there and check and redo the models, excluding some of the references to make it better. Okay, so on the details, that's all that I wanted to show. And then I'll take about 1 minute to show really how to run it. So back to my desktop. So just have a small window here with a terminal where I will open VMD. And I'm going to open Quickfold in the manual mode. I mean, you could open VMD and then load the module. So put the window here, make it big. So far, we only have Alphafold. Model is coming soon. You have to give a single name. So exokinase. So that name is going to be used on the calculation to write the output files. You have to set the path to your installation of Alphafold. So I did it on this folder, on this machine. Your databases, so the huge files, I have it in this folder also. So to give you an idea of how big they are individually, so I'm showing here in the window a snapshot of the sizes. So the big one is the BFD, is the big fantastic database, and then other sequence databases, including one containing PDB structures. So if you go for the full data set, is 2.3 terabytes, but you have the option to use the smaller one that is almost 500. So that really depends on the space you have available and what can you use. So next is the output folder. So I have the exokinase here. And since I showed you, we have plenty of time needed to run. So 15 minutes basically to run the alignment. I'm going to show you actually the result. So the last step here was to load the FASTA sequence, and it will load here as this huge protein. So when you're done loading, you just write the config file to run. And let's pretend I will press fold and wait like half an hour. So we're not going to do that. We're just going to load the models. So quickly, if you set up correctly. So here are 5 models. So in this run, by default, we use all the 5 parameter sets included in Alphafold. So it builds 5 models, and it ranks the models based on the quality. So there's a quality estimate, the PLDDT, and gives them to you. So just a quick alignment. And the last visual step will be, I will show you one way to measure the quality. So I just have a sequence of commands here to edit the structure on VMD. Go back here, I'll just copy and paste them. And as a visual cue, you can investigate the structure by color, and the color is an estimate of the quality. So the blue is a good one, and when you go from blue to white and red, gets bad. So that visualization represents the same that we saw on this map here. So metric of quality. With that done, I conclude my small tutorial on how to use Quickfold, and I'll give the word back to you, Rafael.
Thanks, Diego. Do we have any questions? Yes, we are perfectly on time, and that's a miracle because it's 12:15. So now it's supposed to be the question and answer session, and like we have 15 minutes of that scheduled. If we don't have any questions because we had many questions during the presentations, that's also totally fine. Diego, we can still see your screen just in case.
Hi, Rafael.
Oh.
Yeah, so so there is no such option about selecting like how many models you want, right?
Not yet. Sure. Because it's right now it's standard one default, which is like generate one per database, like Diego was showing.
So just to clarify, the model. So Alphafold generates by default one model. I mean, you choose the method, it will give you one solution. But I mean, it has many parameterizations. So it has many, we call models. So machine learning models. So by default, it has 5 different models. Actually, it's 10. So depending on the scoring metric. So they have the standard and then PTM. So they were parameterized to match different goals on the CASP-14 experiment. So if you do nothing, it will give you one model, but you can select to use with to produce model with different parameter sets. And the parameter sets are called model 1, 2, 3, 4, 5.
Isn't it that like when you select let's say 5 models with your template, with your faster sequence, you want 5 models, so it will give different LDDT scores models. We can choose the best one from the LDDT score.
It is a good practice because you never know which model, which parameter set will render you the best model. So why not using all of them? So there, if you have a GPU, I mean, the part that AlphaFold takes more time really is doing the multiple sequence alignment. When it goes to inferring the structure, it is way faster.
So which GPUs you have used here now?
Can you say it again?
Which GPUs you have used here? Like you showed the timings here.
So the timing is for a A5000. Okay. So it's a top GPU. Yeah, yeah. I think it is equivalent to a 3090.
Yeah, of course.
The RTX 3090.
It is very good.
Yeah, so in the Alphafold website, they recommend you to use an A100, which yes, you know, it's a pretty expensive one, and because we have it, honestly, it's not that different in terms of speed than the A5000. It has more memory, so it could maybe do something larger.
Actually, I was using Google Colab to use Alphafold.
Yeah, so the Google Colab one, they limit the size of the sequence that you can put, but like I think they are using A100s.
Yeah, that is a problem. So in Google Colab, maybe you can run out of memory or exceed the size they allow you, or your session will fall before the alignment ends. So that is really annoying. And maybe, I mean, you have some intellectual property interests that you don't want to share your sequence elsewhere.
Yeah, so these are the reasons why we decided to make first the version that is local, and then work on a version that is going to be somewhat like the Google Colab, but it's not going to be Google Colab. We are going to provide the machines. You know, like that's the idea. Maybe a supercomputer center, things like that. Like not going into something like Google Colab. The other advantage is, like we heard from some other people that wanted to have their local version, like big labs doing MD kind of labs, and they wanted to have their own version locally because for them it would make sense. You know, like, and that's like, but that's kind of our plan right now, like working on that direction.
Actually, this is already available in Chimera X.
Yeah, but the Chimera is the Google Colab.
Okay.
It is exactly the same thing. It's just a browser connecting. It's just a hidden browser inside Chimera connecting to the Google Colab. So there's actually no other tool right now that connects to a local version of it.
Thank you.
So it seems that I have a question in the chat. Somebody is asking, Ahmad is asking if there is any difference in MD engines in terms of accuracy for the same system, the same force field. Excuse me. So I guess at the level that we are usually interested in protein structure, dynamics, and everything should be the same, using different kind of MD engines if you are using the same force field. Even actually between force fields, things are pretty close these days. But of course, in principle, there are sort of, I wouldn't call them inaccuracies. I would say differences between levels of between the force fields and between the level of accuracy you implement, between the tolerance that you implement. Sometimes you make more approximations to make the code faster, to run longer simulations, for example, or you have shorter cutoffs. So the physics is a little bit different. But if you are using the different force field, different MG engines in a standard way, I think you should expect similar results. I wouldn't make a general statement that, yes, I mean, this force field is
I would complement saying if you use the same parameters, right, Imad?
Yeah, it's the same force field, right?
Yeah, if you not just the force field, but like also the MD parameters, like cutoffs and PME and things like that. If you use the same thing, I don't expect any difference. And I also don't expect, I mentioned that to them, I don't expect significant difference also in speed. All the codes are somewhat the same, like in terms of, there are small differences.
Absolutely. Yeah, very small difference in terms of implementing the same physics, you know, what algorithm you use, how you parallelize that, rounding of errors and things of that sort might result in slight differences. Even if you use the same MD engine, the same force field, or the same system, you might get a slightly different answer every time you run it. But those things are usually negligible, so for our business. So Delara, you have a question, you can go ahead and ask. I don't know if it's a question for Rafael or for me.
I think her hand was on, on for some time already. That's
okay.
Now we had quite a few questions during my session too, Matt, so I don't know.
Oh, okay, good. So any other questions also for my lecture? Because I had to rush to another meeting right after my talk. I was already 7 minutes late for that, by the way. If not, then we're gonna break for lunch, right? Rafael, is there something else we need to do for your talk, or no?
That was it.
Okay.
Yeah,
Then we will have everybody back, Mariano, at the same Zoom link at 2 p.m. our time, correct?
Yes.
Okay, sounds good.
In an hour and a half.
In 1 hour and 36 minutes exactly, we're gonna be back in the same Zoom session. Okay, sounds good. Enjoy your lunch or evening dinner or anything you might be doing. See you.
Hi, Dr. Bernardi. Could I ask you a quick question about the Alphafold?
Yes.
So I know based on the installation, you need at least 500 GB of space to install all the files. So for like labs who don't do a lot of MD simulations and they're more wet lab based, like what do you think we should do?
So there is without the VMD, just for the Alphafold, there is a version that is in Google Colab right now, which works for small systems.
Okay.
That's one solution. Like you were the one to talk to me about the collagen, right?
Yes.
Which is relatively large. So
yeah, so the collagen molecule is relatively large, but I'm trying to develop structures for like a smaller bacterial collagen-like protein. So that's only like 80 amino acids long.
That should work, yeah. But like the Google Colab version, it cannot accept much larger files. So what you're working on right now, like would solve your problem, which is provide this as a kind of a service. So like you would have a button there and you would run actually in our computers, not on your own computer.
Okay.
And that's something that we expect to be available in a few months. There are other versions that we are discussing. There are things like AWS from Amazon, which would be actually, in my opinion, great for labs like yours, because instead of buying a computer, you can pay $2 and run the powerful computer, the top powerful computer in the world, for 15 minutes.
Yeah.
Like Amazon offers that service. So that's another way that we are thinking about it. because like we plan to offer the machine here, and that's going to be free, but there might be a queue, there might be a lot of people trying to use it, and we are thinking about alternatives right away, like in terms of if we get too many people trying to use it. So that's one of the solutions.
Okay, I think I like the collab idea, and probably I have to wait for that. So thank you for letting me know.
No problem.